Hi, do you hear me? Yes.
Yes,
Okay, thank you.
now the
I think
sound is good.
it's like a one-to-one session because uh, two to three persons are almost busy and not reply me. And okay, we just start the session too.
Okay.
Okay, thank you for joining us today. As you know, my name is Ali Daraz, and I am thrilled to host this workshop on molecular diagnostics and artificial intelligence. This session will explore how artificial intelligence is transforming genetics screening, next generation sequencing, and precision medicine. Formerly, it's called. In recent years, artificial intelligence has revolutionized molecular diagnostics by improving accuracy, speed, and efficiency in detecting genetic disorders, cancer biomarkers, and disease risks. Okay, now I'm just starting by sharing the screen to you. Uh, First of all, we will talk about by slides. Then uh, we will talk about the websites where we can diagnose about the family medicine problems or other some specific problems by just typing our uh, symptoms, and they will tell us about the symptoms. Oh, sorry, about the disease. Okay. Do you see?
Yes, yes, molecular diagnostic and artificial intelligence transforming healthcare.
Okay, okay, okay. Thank you. Okay. We will talk about today's molecular diagnostics and artificial intelligence transforming the healthcare by Alidras. Okay. Mm, first of all, we talk about myself. My name is Daraz. I am a passionate BS genetics and molecular biology scholar at University of Health Sciences. Brings a wealth of knowledge and hands-on experience in bioinformatics, stem cell therapy, genomics, genetics and evolution, and most importantly, molecular diagnostics. With a keen interest in advancing scientific discovery, Ali is dedicated to inspiring others through engaging learning experiences and fostering a deep understanding of cutting-edge molecular biology techniques.
Yani, are you working at a diagnostic lab or research lab?
I am.
Yeah.
It's a diagnostic lab. If I am sharing to you, okay, I will share you the pics of my lab, uh, but it is in my mobile. But I will send you later.
Okay.
Okay, why genetics and molecular biology? Okay, it's empowerment. Understanding these subjects gives individuals with the knowledge to make informed, health, and ethical decisions. Skill development, global impact, and central dogma. We will talk about, we already talk about that. So we start from the actual session. Okay, molecular diagnostics. Okay, revolutionizing the healthcare with precision and speed. Okay, the word is precision. There is a word is precision. It means this is the point. Always we hit that point. Okay, it means the precise medicine, the precise. Can you off the mic because it's little
Oh, I'm
noisy.
sorry. I'm sorry. I'm sorry.
Okay, thank you. Okay, and speed. Okay, what's the precision? It detects diseases at the molecular level. Okay, DNA, RNA, proteins of the patient. Early detection. The most important diagnosis purpose is early detection. Okay, identifies the illness before symptoms appear. Personalized care. Okay, then we called it now the precision medicine. Is tailors treatments based on genetic makeup. Speed. It's a rapid diagnosis of infections. COVID-19 PCR test. Actually, this is the type of diagnosis. And cost effectiveness reduces the unnecessary treatments because if we don't know about the disease, we will target it from the different ways. But if we know the disease or we know the target, we can reduce the unnecessary treatments and hospital stays. Innovation powers breakthroughs in genomics and drug discovery. Okay. Now, what is the workshop aim? To explore the transformative potential of molecular diagnostics and artificial intelligence. Okay. Uh, this is the formal subject or the type of the program, but we will integrate this by artificial intelligence in revolutionizing the healthcare. Key objectives: understanding the fundamentals of molecular diagnostics. This is the actually the short form of molecular diagnostics and AI. Discover the real-world applications and case studies. Discuss the challenges and future trends. Foster the collaboration and innovation. We will talk about this. Okay, the workflow of molecular diagnostics and artificial intelligence is first of all we talk about the introduction. Then we'll talk about the fundamentals of molecular diagnostics and AI. Then we'll talk about the real world applications. Then we'll talk about the challenges. Then we'll talk about the future trends, and at the end we talk about uh, interactive section in which we will discuss about the session and the what you want to know about the molecular diagnostics and AI. Okay, first of all, introduction. Uh, as we know, the molecular diagnostics utilize the techniques like PCR, NGS, and microarrays. I hope you know about the PCR. PCR is actually uh, is a technique in which the gene of interest is amplified. Okay, amplified is exponentially. Okay, when we amplified it, then can use it. We can use it for sequencing and other techniques. And the next thing is NGS. It is the most latest form of the sequencing, that is next generation sequencing. And then at the end is the microarrays. Actually, the macroarrays is simultaneously know about the genes. Okay, identify the genes simultaneously. Uh, it is the speedily a technique you can say, in which solid, it's like a solid chip on which the cDNA is hybridized here or bind here. Okay, it's like a pores or wells on which the cDNA is bind, and then we bind the probes. Okay, probes are actually the single-stranded specific nucleotides that are complementary to the cDNA, and the fluorescence dye is attached with the probe. When we hybridized with the cDNA, and when we check in the fluorescent uh, machine, uh, the gene which is complementary to the probe is fluorescent, is giving the color, and the gene which is not complementary to the probe is not fluorescent and not giving the color. By this we can identify the target gene. Okay, and to detect the genetic variations. Okay, if there is any variation or the mutation, we can detect it. Enables the target therapies based on genetic makeup. Okay, if the patient has a different genetic makeup than the community, okay, and we give the same medicine as we are giving to the community, it will not help that patient. Okay, then we have to enable the targeted therapies based on the genetic makeup. That is actually called the precision medicine. Enhances the accuracy, speed, and efficiency in genetic analysis. Okay. Okay. Understanding AI in genomics or in the molecular diagnostics. AI versus the traditional methods. Traditional methods rely on manual analysis and statistics. Okay. They are relied on the manual analysis. The techniques is same as we do the PCR, we're doing the sequencing, but the interpretation is very difficult, okay? And it is very subjective. But if we interpret interpret the result by AI, by artificial intelligence, it is very it is very precise and it will be done very speedily. Okay, the deep learning and machine learning, it is actually the languages that is used in the bioinformatics. AI models learn from the genetic data. Okay, actually they work on data. Okay, the more data they consume, they will become the powerful. Okay, AI models learn from the genetic data to make the predictions. Applications in molecular biology, AI is used for gene expression analysis, mutation detection, and protein structure prediction. Okay, gene mutations and AI. As we know, there are the two types of mutations. Okay, first of all, it's gene mutations, and the second one is the chromosomal mutations. Okay, the second one is the chromosomal mutations, and we also called is uh, genomic mutations, the genome mutations. The types of gene mutations are the point mutations in which just the one nucleotide or the base pair is changed with the another. Insertion in which the amino acid or the base pairs are insert. Deletion in which the base pairs or the nucleotides are delete, and frame shifts. Okay, we will talk about the frame shift. Actually, as we say, it's A T G C C G. It's like a you say it's a strand, sorry, it's a DNA strand having the nucleotides. Okay, as we all know, the amino acid, one amino acid is based on is made up of the three nucleotides. Okay, so we can say there are two there are two amino acids. Okay, there are two amino acids. But if the one, for example, this T, the is insert the T is deleted. Okay, if the T is deleted, it will become A, G, C, C, and G. Okay, now the three codons are that, and that one is incomplete. Okay, and now the amino acid is changed. The amino acid is changed. So by frame shifts, the amino acids are changed, and this is called frame shifts. Okay, AI for detecting the mutations. Okay, now we'll talk about the AI that are detecting that type of mutations, like the gene mutations. AI tools like deep variant, we'll talk about on the websites, and the mutate 2. There are two types of tools that identify the mutations with high precision. Okay, and if we talk about the case studies, AI in mutation analysis is AI assisted genetic testing in hereditary diseases. Okay,
Sorry علي, the tools, it's like a program يعني, the deep
which
variant,
which one?
the tools, the point before
This is
AI for
deep
detecting
variant.
this with software program. What is it?
These are these are actually the tools. Okay, we will talk about in the session is the deep variant
Okay,
is like a tools in which we can identify the mutations. Okay, first, for example, we have a sequence of a patient. Okay,
okay,
NGS
for
sequence.
sequencing يعني. Okay, for sequencing
Okay, okay,
data analysis for sequencing.
yes. If we have a sequence
Okay,
of the patient, we will upload in deep variant or a mutate too. Okay, and then we they will uh, uh, match with the reference. Okay, and they will tell us the where are the mutations are actually,
okay,
and
okay,
what is actually the uh, effects of that mutation.
okay.
Okay, now we'll talk about the second type of the mutations, that is chromosomal mutations, in which the deletions, the duplications, and inversions. And the deletion means the part of the chromosome is deleted, and the duplication in which the part of the chromosome is duplicate, and in the inversion, the part of the chromosome, for example, this is the chromosome, for example, in which this part is excised. Okay, this part is excised, and one part is like that, and second part is like that. Okay, in which it is flip out. Okay, it is flip out. It's like that. It is flip out and reattach with it. Okay, that is called inversion. And in the translocation, the part, for example, this is the one chromosome, this is the second chromosome, in which the part of the chromosome is the one chromosome is exchanged with the other chromosome. That is called the translocations. And AI for large scale genomic variants, in which the AI helps they detect the copy number variations. The copy number variations, it means the the number of genomes in the one individual is actually called, or in the one cell of the individual is called. Abnormalities like translocations, inversions, duplications, and deletions can be identified by AI. We'll talk about later by the tools. AI tools used in the analysis is AI-powered karyotyping and CNV analysis.
Ali, sorry, I didn't understand
Uh,
yeah, the last one. Copy number of variants.
okay, okay. Actually, okay, okay. I will talk about. Actually, in our genome, there are
Yeah.
the in our genome there are three types of uh, there are three types of we will talk about the first of all there are introns.
Yes.
Do you know about the introns and exons?
Yeah, yeah, yeah.
Okay, introns and exons,
Exons. Yes.
and the third one are the repeats. Okay, in our genome there are three types of things: the introns, the exons, the non-coding part, and the repeats. Okay, the repeats, the one type of the repeats are tandem repeats.
Yes.
Okay, the one type
Repeats.
of the repeats are tandem repeats, in which in tandem repeats The nucleotides like the A, T, G, C are are in the pattern like head to tail. They are in the head to tail. It means there is a chain of the repeats like AT and then again AT. Okay, and
Okay.
that are repeats are like the thousands of the repeats 80, 80, 80. Okay, the sequencing of that repeats are very difficult. Okay, the shotgun sequencing, the NGS, it's very difficult because the software is get a problematic to analyze or the sequence that type of things. Okay, by this, if the number of repeats in one individual is like 50, for example. Okay, in one individual, the number of like the repeats are 50, and the other individual it is 52. Okay, do you understand?
Yeah.
Okay, by this there is a polymorphism. Okay, because
O-
in the one population, the number of repeats are different in the different individuals. Okay,
Okay.
that is called the copy number variations. Okay, that is
Okay.
the number of repeats in the one individual is different than the other individual. Okay,
تمام.
that are
Okay.
that are the that are the CNVs, and the one thing is the SNP. Do you know about the SNP? Is the single nucleotide polymorphism, in which
Yes.
in which the just a one in which the just one like the T, the one nucleotide is changed in one individual, and in another individual it is different like a. In another
Yes.
individual it is like A, and in another individual it's like a T. So it's called a single nucleotide polymorphism.
Yes.
Okay. Do you understand it? Now
Yes.
we'll talk
Yes.
about the next one. Okay. Okay. Now the AI-powered karyotyping and CNV analysis are equipped in which do you know about the karyotyping?
Yes, I know karyotyping.
Yes, in which uh, in karyotyping on the basis of size and and the centromere position and the centromere position we arrange the chromosomes and we check the mutations or the deletions or insertions in the chromosome level. We will identify the effects. Okay. Now we'll talk about the NGS. We already have a good workshop on have a session on the NGS, but we'll talk about on the basis of the artificial intelligence that is equipped in NGS. Okay, the NGS. It's a high
Yes.
throughput. It's a high throughput sequencing technology for entire genomes. Okay. In the previous ones, the Sanger sequencing, it is very low throughput. Okay, it will take almost the 1 month to years to sequence the whole genome. But by the NGS, it will be done in the days or the weeks. And the steps in the NGS analysis is like the first of all we have to prepare the library in which we fragmented our genome into 200 to 500 to 1000 uh, fragments. Then we sequence it. There are different types of sequencing. Uh, the one approach is Illumina, that are actually the NGS platforms. First of all, it is Illumina. The second one is the Nanopore. Third one is the uh, I am just iTorrent. iTorrent. These are types the actually the platforms that doing the sequencing. Actually, the more specifically NGS sequencing. Okay, and then at the end, the data analysis. Actually, AI is doing the data analysis. Challenges and AI solution. there are a lot of challenges in the data analysis. Okay, and the solution is AI. AI is optimizes the read alignment, variant calling. Variant calling means if there is any variant in the sequencing, and the annotation. In the which annotation they will, because every mutation is not cause any effect. Okay, every annotation doesn't cause any effect. Okay, some mutations are like the silent mutations that doesn't change the amino acids. Okay, and some mutations are like the substitution mutations or the conserved mutations that change the amino acid, but the changed amino acid are having the same physiochemical properties. Okay, so that amino acid doesn't cause any effect or the disease. Okay, so we have to remove or the filter that type of mutation, that the silent mutations. So we have to do the annotation. That is also done by AI. Okay, the AI in NGS. AI detects SNPs. Okay, as we all know about the NGS. Indels. Indels is actually the insertions and deletions, and the structural variations. Structural variations means the deletions, the insertions, the inversions, and the duplications of the chromosomal part, and also the translocations. It's actually the structural variations. AI-powered algorithms like the deep learning models improve the sequencing accuracy. AI-assisted NGS for rare diseases diagnosis is actually the case study in which the rare diseases can be diagnosed by NGS. Okay, now we'll talk about. Sorry. Okay, okay. The convergence of molecular diagnostics and AI, in which the precision medicine is a one type of the uses of the AI, in which AI enhances the molecular diagnostics, enabling the precise individualized treatment strategies. Okay, in which what we will do in the precision medicine, in which first of all we check the genetic map makeup of the individual. Okay, when we check the genetic makeup, then we prescribe the medicine to that individual. That is actually the precision medicine. Okay, it is the shortest definition of the precision medicine. The next thing is data-driven insights. Okay, first of all, they get the data. Okay, then they annotate it. Okay, they filter out it, and then we get the actually the what which mutation has what type of the effects. The molecular data combined with the AI algorithms to generate the powerful clinical insights. Okay, as we all know about the PCR, it's actually amplifies specific DNA sequences for detection and analysis. Okay, as we all know about the PCR. And the next generation sequencing, in which it rapidly sequences the entire genomes of target diseases, as we will talk about. Okay, the fish, in which the fluorescence in situ hybridization, in which the fluorescence is hybridized with the complementary part of the chromosome inside the cell. So we don't have to remove the DNA, or that if we don't have to extract the DNA and then uh, further do the study. But in fish, we didn't do the extraction. Visualizes the specific DNA or RNA in the cells or tissues. Okay, we don't have to remove the or extract the DNA. Okay, AI fundamentals for the healthcare is the first of all, it's a machine learning. Algorithms learn from data to make the predictions or the decisions. Okay, the neural networks, complex algorithms modeled after the human brain. It's actually the about the neuroscience. And the computer vision enables the AI to analyze and interpret the images. Like as we all know about that, there are different images like the radiology, in which X-rays or the different types of scans can be interpreted by using the AI. And the AI powered cancer diagnostics, in which the image and analysis. Okay, as we scroll the LinkedIn newsfeed, in which we get about that, the image of this image of the skin, in which the AI actually tell about where is the uh, cancer is developing. It's all done by AI. AI analyzes the medical images to detect these subtle anomalies. Anomalies means abnormalities. And the genomic profiling, AI integrates the genomic data to predict the cancer risk and progression. Do you hear me?
Now, yes.
Okay, okay,
Before it was.
okay, okay. Okay. And the personalized treatment, as we all know, the precision medicine, in which the AI guides the treatment decisions based on the individual patient profiles. Okay, AI infection diseases. Okay, we are talking about the gene mutations, the genome mutation, the chromosomal mutations, the molecular oncology, and now we'll talk about the AI in diseases, infection diseases. The pathogen detection, it's also done by the AI that rapidly identifies the infections and agents from the clinical symptoms, samples. Sorry. The resistance prediction, AI predicts the antibiotic resistance to guide the treatment. And the outbreak monitoring, AI tracks and predicts the spread of the infectious diseases. Okay, the ethical consideration, in which we use the AI in the molecular diagnostics, but there are the ethical considerations like the bias mitigation. Okay, AI may be biased. It's actually a big problem. The data security. We give the data to the AI, but it may be insecure. Okay, that data may be insecure. It will be uh, cost very much high because maybe the handlers of the AI can sell it to someone else. The transparency. Uh, AI is not like a transparent. It's not like a glass because, as we all know, AI is doing the different types of algorithms behind us. Okay, so it's not a transparent. The ethical considerations are important to mitigate the bias, protect data, and ensure the AI transparency. And challenges and limitations like the data quality. If the data is very low, the interpretation is very weak. The interpretability, it's like the ability to interpret is very low. And the regulations, because the government and the other types of agencies that regulate the AI uh, maybe have some weakness. Okay, so the regulations are very poor. These challenges need to adjust to ensure the responsible and effective use of these diagnostics. Okay, now we talk about the emerging trends like the liquid biopsies. As we all know, as we all know, the cancer organs release some types of nucleic acids in the blood. Okay, if we diagnose the blood, okay, like the cell-free treatments or the this type of techniques, we can diagnose the cancer without doing the surgery. Okay, so this type of operations or the biopsies is called liquid biopsy. AI-driven drug discovery, it's also a emerging trend, and the wearable diagnostics. Okay, we will wear the watch and we will uh, check our heart rate. We will check our sugar level. We can check our the breathing speed by using the variables. Okay, that are also the emerging trends. And embracing the potential, the molecular diagnostics and AI offer transformative opportunities. This collaboration can reshape the healthcare for the better, for the betterment of the community or the individuals. Data requirements. This is actually the, you can say, okay, data, if the data is available, the AI is good, AI is best. If data is not available, AI is nothing. Data diversity. Diverse and representative data sets are needed. Okay, diverse, the different type of data, and representative, that represent the individuals, that represent the population, that represent the community. That type of data sets are needed for training the AI models. Okay, if we lack that type of data, AI is nothing. AI is doing nothing, that type of things. Data labeling. We have to label the data. That data is from Asia, that data is from the Africa, that type of data is from the Europe. If we label the data, then we can supervise the AI modeling. Okay, the accurate data labeling is crucial for supervised learning algorithms. Okay, so the labeling is very important. Data accessibility. Okay, if we didn't access the data, we can't interpret we can't interpret that type of data. Okay, so public databases like NCBI and TCGA provide the accessible data sources. So we have to explore that type of nucleotides, NCBI and TCGA. Okay, okay, now we'll talk about some type of, okay. So the one, okay, so it's actually a website. Do you see that website? Yes. Hello. Do you hear me?
Yes, Ali, I'm hear you.
Okay. Okay, Miles. Now we will talk about the websites. Typing our problems or the symptoms, and the AI will talk about will tell about the disease or the effects. Okay. So first of all is the docus. Okay. Now if we try it, just try docus for free. It's a free website. Okay. Continue it. Okay, just remember that type of websites because you can also explore it later. It's like, okay, okay, now it's start. Okay, first of all, We will tell about our age. It's 22 to 23 years. And talking about the, okay, it's 155, height is 5'8", and it's a South Asian. It's a male. Okay, which Docus AI feature would you like to explore? Okay, we can select the personal AI doctor, the lab test interpretation, the consultation with the doctors. Okay, as we talk, easily understand and interpret your lab test results, we can select it, lab test interpretations. Okay, we add by upload or the enter manually. We just enter the manually. Okay, first of all, we check the hemoglobin level or that type of things, in which they enter a summary of your screening results, including the key findings and the measurements, like the hemoglobin level is 8. Okay, we just making as something like the results or the this type of things, and I am feeling ill, fever, this type of things. It's a physical examination. Select date. We just check the date. Additional no. Okay, this type of things. Screening. Okay. It's just a uh, it's paid. Yes, it's paid. I just try it. It's free, but now it's just asking about the payment. I think so. We just skip it. We will talk about it later. First of all, we talk about that My Medical, in which they check your symptoms, in which we just uh, first of all we agree with it.
How may I help you?
Okay, that is first of all we talk about like the headache. Okay,
هذا this is another program, yeah?
Please
Different.
indicate your gender.
مش دوكس,
Please
not
indicate your age in years.
دوكس, صح؟
okay, I am not. Uh, yes, now yes.
This is
Do you hear
another
me?
one, صح؟
Tell
Yeah,
me
yeah, I
what
hear
are
you.
you asking about?
The program, this is another than دوكس.
Sorry?
هلا, this program, this one at this screen is another one, not same as دوكس, yes or no?
Yes, yes. That is the second website. The My
Yeah.
Medic. Yes, it's the second.
What is name?
Okay. I am sh sharing to you or just
Okay.
Please indicate your gender.
okay. First of all, we just select our gender.
Please
Then we
indicate your age in years.
okay. There's a 22.
Could you please
And
indicate where you are? Adding your location will improve the accuracy of the result.
okay, okay. Yes, it's Lahore, Pakistan,
What signs or
and
symptoms do you feel?
I am feeling headache.
If you are in
The
pain, state the painful areas.
yes,
Otherwise, please continue.
headache.
Please specify in which of the head and or neck areas you have pain.
Yes.
Indicate which region of the world you have visited in the last 8 weeks.
I am from Asia.
Please state the intensity of your pain using the number scale,
It's a
ranging from have you
moderate.
had a fever or feeling of
No, no, no.
do you have neurological symptoms such as mouth deviation, loss of do you
Is
have sinus pain?
no.
Are your
Are your
headaches throbbing?
headaches throbbing? No.
Did the
Did the
headache
headache
start while
start
you were
by?
doing intense physical
Okay, I am just
Can you take
uh,
have you traveled to India during the
getting the
last 8
result
weeks?
as soon
Please
as
indicate
possible.
how long ago the symptoms
Yes, it's
started.
like hours.
Please indicate how many hours
Okay,
ago the
it's like
symptoms
1 hour
started.
before I am feeling the
Since the
headache.
onset of symptoms,
Good
how would you say your
progression? No,
condition
it's a
has
poor
evolved?
progression.
Is the headache mainly behind the
Headache
eyes?
mainly behind the
Have you
48
consumed
hours.
alcohol in the last 4? Do you
Drink
drink
alcohol?
alcoholic
No, no.
beverages most?
I am Muslim.
Is it possible
Is
that
it
you have
possible
been
that you
bitten by a
have been
mosquito in the
bitten by the
last month?
mosquito? No. Okay. Now we get the result. Okay. In which we talk about the different type of things. That's actually the now that's the result. Okay. It's a low urgency. Okay. So we don't have need to go to hospital. Okay. Check your case with the health professional, and if your condition worsens, go to hospital. But the urgency is very low. Okay. The color is green. That is the actually the color. If the color is red, it means that there is a urgency and you have to go to the hospital. And it's a common headache. It's a select it's just like the low match and the specialty. If you want to go to the hospital, you should go to the family medicine. Okay, you don't need to go to the internal medicine if the match is very low. Psychologist, you have to go to psychologist. No, you don't need emergency medicine. No, it's a very low match. Neurobiology, neuropsychology is very low match. But the family medicine is a very high match. So you have to visit the family medicine doctor. So this type of AI is in which okay, it's a that type of things. And in ADA Check Your Health is actually a app. You can uh, you can download this app, install on your mobile, and just typing your symptoms, and they will tell about the effects of the disease, or you have to visit the doctor or not. They will tell you. Okay. Now, this is actually in which we can schedule the meeting because the servers are very slow. So you have to schedule the meeting for the 30 minutes or the 80 minutes in which you can schedule it. It's, we will share the links of that all type of the websites to you later after the okay.
to 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 to, to apply the genomics lab يعني NGS whole exome whole genome and in the future we will do more يعني more tests but I need يعني to build basic at NGS.
Okay,
You
okay.
know, when you work you have
Okay.
يعني
Okay.
to
Did
learn
you
to
did you
yeah.
take the NGS workshop that I was take a session about the NGS? Did you take it?
Yeah, yeah. This is the first one I will take it, the NGS.
Oh, okay, 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 okay. Okay,
But
and did you familiar with the bioinformatics?
هلا for now no صراحة. حتى even when we do the validation we will do the يعني we will do the technical works at the lab. At the first stage يعني we will يعني after work we will send the data to do bioinformatics by third party
Okay, okay.
ف ف ف
Actually,
for
actually,
me I had there is online session I يعني I will I will attend يعني to build basic information about bioinformatics and
okay.
NGS.
Actually, the bioinformatics is a fundamental of the, you can say it's the basics of everything. Okay. I'm not saying, even don't, we can say the basics. It's actually the interdisciplinary subject that is very crucial, very important. Because if we don't know about the dry lab, it's actually we call the bioinformatics is a dry lab. If we don't do the dry lab, we don't get into the wet lab. That is actually the molecular diagnostic labs or the types of things. And did you ever do the PCR? Okay.
PCR, yeah. We have PCR, we have digital PCR.
Okay. For PCR, we have to arrange the primers. Okay.
Yeah.
Okay, and we have to design the primers. Okay, so the primers are designed by bioinformatics. Okay, so
Yes, yes.
bioinformatics is very important, and we have to do it.
Yeah.
If you do you familiar with the primer designing? Can you do it individually without any help?
Yeah.
You can do by using the primer tree or
Sorry, I didn't hear the question
oh,
very well.
I am just asking that can you can you design the primers without individually without help getting help from other?
I didn't did it before. Truly, I didn't did it before because we are زي ما قلت لك we are a diagnostic lab, and we buy all the kits, CE IVD had because we are CAB accredited lab. فأصلا we have to buy um, ready kits that have certificate like CE IVD.
Okay, okay. So
ف
it's a
I never do it before, you know,
okay, okay, okay.
to
It means
design primer.
it means we have to do you want to learn about the how to design the primers or
Yes, yes,
and
I'm
everything.
interested. I am.
Okay, and everything about the bioinformatics, because I
Yeah,
am really good in bioinformatics. Okay, and if
yeah.
you
I will
want,
share you. I will share you link. I found it at LinkedIn. There is course. I think it's basic, and it's online. I will share it with you. يعني I'm thinking in to start with it.
okay, there are different types of courses also available on the Coursera. Okay, that the
Coursera, yeah.
yeah, so bioinformatics one, bioinformatics two, that type of
And
Coursera.
it's useful for basic level, yeah.
Yes, just a basic level, okay. But
Yeah.
if you want that type of things that exploring the NCBI, Ensemble, Uniprot, the PDB, and the multiple sequencing by doing just checking the cluster omega and using do you know about the phylogenetics trees?
The pharmacogenetics, the pharmacogenet.
Phylogenetics tree, phylogenetics trees.
No.
Okay, that's also you can say the crucial technique for phylogenetics tree. The relationship between as we all know, we if we just don't assume we are Muslims, because if we are Muslims, we know we are we are made by Allah. Okay, but if we talk about the science, okay, they tell us we are closely related to the chimpanzees. Okay, how will we
Okay.
get it? Okay, how will we get it? We know about it by using the phylogenetics trees. Okay, because the branches of the taxons are very close. The Homo sapiens and chimpanzees, the humans and chimpanzees are very close in the phylogenetics. Okay, that all
Okay.
are done by the bioinformatics. Okay, and that is done by using the bioinformatics softwares like the Mega. Okay, if
Okay.
you are if you ever tried to learn the bioinformatics in the one-to-one sessions like that, we can also do that. Just
Yeah, yeah, yeah.
tell.
Yeah, we can do it.
Okay, so for this we don't need to make the slides. We just make a session, okay, on the
Okay.
weekends, okay, and we will explore the different types of databases, and
Okay.
and we will also talk about the different types of softwares in which we can do the phylogenetics trees and the 3D structures of the proteins in which if there is any mutation in the proteins at the protein level, the structure of the protein is changed. Okay, so we also explore that type of things. So you will definitely uh, get the knowledge about the 3D proteins and the multiple sequencing alignment. Okay, as we all know, the NCBI, the blast that is the basic local alignment. The word is local. It means there is a matching between the very close sequences. Okay, but there is a multiple sequence alignment in which we sequence the different types of sequences. Okay, so like the 20 species, the 30 species can be sequenced. That's also done by multiple sequence alignment. That's also done by bioinformatics.
Yes.
Okay, inshallah, we will do it in the next sessions. Just
Inshallah. Inshallah.
just message me. Okay, I will save it and we will do it in later. If you
Inshallah.
want, okay, if you want a session tomorrow or today night, I will also arrange it if you
Okay.
want.
I prefer to schedule مثلا once a week like Friday at 7. It may be the best time because I have only one day um, off. It's Friday. فmaybe we arrange every Friday we will do a session in a variant time because برضه now we have رمضان كريم يعني ف
Yes,
there
yes,
is
it's
there is
it's just
there
started.
is special
To, today's
schedule
today's
for رمضان يعني.
today's Ramzan is started or from tomorrow?
Oh, okay. احنا already start يعني today is the first time first day of Ramadan.
Okay, okay. In Pakistan, it's just started tomorrow.
Tomorrow.
Yes, it's
يلا
just
رمضان
started
كريم.
tomorrow.
Mm,
Thank you, Ramzan Kareem Mubarak.
thank you. Thank you. خلاص.
Okay.